Hello, today we start to study immunology, and this is our first lecture. You see the topic infection, pathogenicity and virulence, immunity, and factors of non specific resistance. I think it's too much for one lecture, and maybe I divide it into two parts. And here's a lecture schedule. We start with infection, its definition, differences between infection process and infection disease. Mm, We will study the form and stages of infection process, pathogenicity and virulence, and factors of pathogenicity of microorganisms. And I think this material is enough for first part of our lecture. And the second part will be about the immunity, its definition and types of immunity, and about non-specific or innate or inborn immunity, which includes barrier function of the skin and mucous membranes, bactericidal substances of saliva, which is very important for stomatology students, and some cellular and humoral factors of non-specific immunity. So what is it infection? Infection is invading of the microorganism into macroorganism and its multiplication inside host organism, which is resulting in infectious process. Infectious process is an interaction between micro and macroorganism under certain environmental condition. It can develop as infection disease, carriage without clinical symptoms, or vaccine process after injection of vaccine. Infection disease is the most pronounced form of infection process. Infection disease have many differences from the somatic ones. They cause it with live agent, live causative agent, which is maybe bacteria, virus, protozoa, or fungi. Infection disease have incubation period, which may last from some hours for some months. And the immune response develop as a result of interaction between the pathogen and human body. And infection disease is mostly contagious. They can be transmitted from person to person in different ways. At this slide, you see the list of many types of infection according the causative agent, the entry portal, the course of infection, mm, the spread of bacteria and viruses in microorganism, according the number of infection agents, and according the development. And this is a part of your independent work. Please find the definition of all these types of infection, and it will be controlled and be checked in our practical classes. This independent work is for all students. The infection process develops in some stages, and here you see the list of the stages. And this material is not very difficult to study independently, and you have one more piece of independent work. Please, in your copybooks, briefly describe each stage of infection process, and this material we also will check in practical classes it next week or this week. And which factors affect the infection process? It's the interaction between host and pathogen in some environmental condition. Factors associated with host are um, the age, sex, the state of immune system, the type of nutrition, mode of life, the occupation of people, all this um, important for development of action process. Environmental factors include the climate and the industrial pollutions in environment and social factors also. The development of infection process depend on all of them. And the factors associated with pathogen, it's presence of some virulence factor in it, and also um, the ability of this pathogen survive in environment, the entry portal and infection dose when this pathogen invade the host organism, and all this important in development and infection. But in microbiology, we paid the special attention for the factors associated with the pathogen, its pathogenicity and virulence. Pathogenicity is the potential capacity of certain species of microorganisms to cause infection process. It's a genetically determined and inheritable property of some species of bacteria. Some bacteria are pathogenic, so they cause classic diseases. Others may be occasionally pathogenic, they cause disease only in immunocompromised people with weak immunity. And many bacteria are non-pathogenic, they never cause diseases. The virulence is degree of pathogenicity of a given culture or strain of microorganisms, and its index of the qualitative individual nature of the pathogenic microorganisms. Virulence can be changed under the influence of environmental conditions. We can measure the virulence, and we have special units for measure. It is the DLM, or dosis letalis minima. It's the minimal amount of live microorganisms which kill 95 to 97% of infected laboratory animals in a certain period of time. Other unit is the DCL, dosis cetralitalis, the absolute lethal dose of pathogenic microorganisms which kill all infected experimental animals. But mostly we use this indicator is LD50, the dose which is lethal to 50% of infected animals. And this indicator mostly used to compare the virulence of different bacteria or viruses. The virulence of pathogenic bacteria associated with their property for adherence, invasiveness, capsular production, which protects them from phagocytosis, their aggressiveness, so the ability to suppress the immunity, and toxin production. And these virulence factors may be present in bacteria in different combinations. The first step of infection is mostly attachment of bacteria for the mucosa or wound surface, and some factors activate this process, like filamentous hemagglutinin, which cause the adhesions to erythrocytes. It's present, for example, in causative agent of whooping cough. Fimbria, mostly present in gram-negative bacteria, they help to them to attach to the solid surface. Glycocalyx or capsula mostly inhibit phagocytosis, but also participates in adherence. Slime, which is larger than capsula, and it participates in the production of biofilms, which are very important as an adhesive factor. And teichoic and lipoteichoic acids in gram-positive bacteria also serves for the attachment to the host cells. And here you see the electronic micrograph illustrating the adherence of staphylococci to the epithelial cell. And here you see the mechanism of adherence. Bacteria have fimbria or lipoteichoic acids like gram-positive microorganisms, and they interact with some receptors, some molecules, and the surface of human cells. Very important virulence factor of bacteria is a capsula. Capsula makes them resistant to phagocytosis and antibodies, and increases their invasive properties. For example, in the species Bacillus anthracis is causative agent of anthrax, only capsular bacteria are pathogenic. They can multiply inside phagocytes, while non-capsulated variants are not pathogenic, and vaccine strain is non-pathogenic Bacillus anthracis. Some pathogenic bacteria like Bacillus anthracis, Clostridium perfringens, or Streptococcus pneumonia produce capsula only in human body or in the animal. But others, like Klebsiella, produce capsula in all occasions in organism as well as in nutrition media. And here you see the capsulated bacteria with special method of staining, it's buridin staining. Invasive properties of bacteria is their ability to penetrate tissues of the infected organisms. And bacteria possess some enzymes which source for invasiveness. Firstly, it's collagenase and hyaluronidase, which degrade collagen and hyaluronic acid respectively, and thereby allow bacteria to spread through submucous tissue, like you see in this diagram. Immunoglobulin A protease degrades immunoglobulin A at the surfaces of mucosa where it's normally present, and in this way allowing the microorganism to adhere to mucous membranes. It's produced by many bacteria like Neisseria gonorrhoeae, Haemophilus influenza, and Streptococcus pneumoniae. Leukocidins, the toxic substances which destroy neutrophils, leukocytes, macrophages, all these phagocytic cells in human body. M protein of Streptococcus pyogenes also possess the antiphagocytic properties, and the protein A of Staphylococcus aureus can bind to immunoglobulin G, the antibodies, and prevent the activation of complement system. Other important enzyme of invasiveness is the coagulase, produced, for example, by Staphylococcus aureus. It leads to formation of fibrin clots in blood vessels, and also this fibrin layer protects uh, bacteria from phagocytes, from antibodies, and the source for mimicry inside human body. And in this diagram, you also see the two types of invasion of bacteria. Some of them invade the epithelial cell, and other can spread between the cell in intracellular spaces and reach submucosal layer, and then may spread in other tissues. Very important pathogenicity factor of bacteria is the toxins. They divided into two large groups, exotoxins and endotoxins. And this table illustrates the difference in their properties. All exotoxins are proteins in their chemical composition, while the endotoxins are lipopolysaccharides. Being proteins, the exotoxins hit labile, so they easily destroyed at high temperature. Exo means they're actively secreted um, from the live cells in the surrounding medium, and many of them have properties of enzyme, and they're very specific in their tissues. Some of them neurotoxins, other cytotoxins, and maybe enterotoxic, and the effect is very specific. Endotoxin as a lipopolysaccharide, so they heat stable. They actually it's a part of cell wall, the automembrane of gram-negative bacteria, and they released only after cell death. They have no properties of enzymes. The action is non-specific. The endotoxin of different bacteria have the same mode of action. Exotoxins are very powerful poisoning, much more toxic than other biological toxin like poison of snake or spiders. Um, Very small amount of it is lethal for human. They are highly antigenic, so they activate the immune system to produce antibodies, and these antibodies, the antitoxic, they can neutralize toxin and doesn't affect the bacterial cell. In the special treatment with high temperature and formaldehyde, they may be converted into some type of vaccine named toxoid. And endotoxins produced with gram-positive or gram-negative bacteria. Endotoxins are not so toxic like exotoxin, and they have weak antigenic properties, some of them antigens, but they don't stimulate the formation of neutralizing antitoxic antibodies. They cannot be used for production of vaccine like toxoid, and only gram-negative bacteria possessing outer membrane with lipopolysaccharide produce these endotoxins. And here you see the examples of the most important exotoxins. Exotoxins, and you see the name of bacteria producing them and the type of action of these toxins, some of them neurotoxins, either other cytotoxins or enterotoxins. And this is disease caused mostly with action of toxin, not bacteria, and mm, the mode of action of these toxins. And this slides also illustrate the specificity of exotoxins. And for example, tetanus toxin blocks the release of inhibitor mediator, and it results in spasm and seizures, which is characteristic symptom of tetanus. Staphylococcus aureus produce alpha toxin, which is a hemolysin. It causes lysis of erythrocyte in blood agar like this, and in human body also. And Vibrio cholera produces enterotoxin, which serves as adenylate cyclase, and, and under the influence of this toxin, the concentration of cyclic IMF increase, and it leads to release of excessive water in the lumen and the watery diarrhea. So each exotoxin very specific in its action. Endotoxins have similar action, and it's not dependent on the uh, species of bacteria. And here you see the chemical structure of this toxin. It's a part of outer membrane of gram-negative cell wall in gram-negative bacteria. And the common symptoms caused with endotoxins is the fever, maybe blood clotting, and hemorrhages in small blood vessels, inflammation, and hypotension. In severe cases, if a high amount of endotoxin is released into bloodstream due to fast death of bacteria, destruction of their cell, it may cause the coagulation of blood in the blood vessels and even death. And if you have any questions about this part of our lecture, about pathogenicity and virulence, ask me by email. And our second part of lecture, it will be about immunity definition and all mechanisms of innate immunity in people.